Hello, and welcome to our final project presentation video. In this video, we'll be discussing whether Lake Murray is safe for recreational use and analyzing whether its impaired status under Section 303D of the Clean Water Act is reflected in analysis of fecal coliform concentrations. This project is for Environmental Engineering 363, and the group behind the project are Dana, Miriam, Michelle, Daniel, and Khalifa. Let us begin with the lake itself. Lake Murray is a great recreational and aesthetic resource for the neighboring communities. It's a popular destination for shore fishing, private boats, walking, and picnicking. The objective of this project is to determine whether Lake Murray is safe for recreational 2 and recreational 1 use, which pertain to recreational water activities that involve proximity to water, but also the likelihood of contacting or ingesting water. This will be determined by sampling and analyzing presence of fecal coliforms and determining whether they are compliant to the Water Quality Control Plan for San Diego Basin regulations. The codes and standards in San Diego for recreational 1 and recreational 2 are that fecal coliform concentrations shouldn't exceed 2,000 per 100 milliliters, and that E. coli shouldn't exceed 126 colonies per 100 milliliters. Given that Lake Murray is listed as an impaired body under the Clean Water Act, it's anticipated that it will contain high concentration of fecal coliform and E. coli, either borderline or potentially exceeding maximum requirements. The purpose of this laboratory was to test this hypothesis by sampling surface water at Lake Murray and testing for these parameters.
Now into our methods. As part of our presentation, I'm going to go over experimental and statistical. So our water samples were collected on the east side of Lake Murray. We did 2 sampling days, one on April 12th and one on April 18th. So for our water samples, we did 3 grab samples, each using sterile plastic containers. We opened and closed immediately after collection and try to avoid any contact with our samples to avoid contamination. And in terms of dilution, we first hypothesized concentrations would exceed 2,000 ppm per 100 milliliter. So we concluded that dilution should be 1 to 10 and 1 to 100 for analysis. And next, for incubation, we incubated our samples for 16 to 20 hours. And in terms of actual testing, we analyzed for total coliform E. coli using the IDEX coliform test with the Quantitrade 2000 system. The system detects 1 to 200 organisms per 100 milliliter, and it delivered automated quantification methods based on the most probable number model. And next is the statistical methods. We used the t-test to compare our data, and then for illustrating our data, we used bar graphs for each sample, and averages were depicted of each days and together over both days. And we included 2 pictures of our testing sites for day 1 and day 2.
Both sampling groups are shown to be the most accurate at the 1 to 10 dilution level, given that the 1 to 1 and 1 to 100 dilutions resulted in concentrations outside detection limits, as depicted in tables 1 and 2. Results of total coliform and E. coli concentrations from the Quantitrade 2000 of Lake Murray proved to be exceeding regulations and under impaired status. The average results were shown to exceed regulatory limits, with an average total coliform concentration of 4,809 mpn per 100 milliliters and average E. coli concentrations of 918.5 mpn per 100 milliliters. However, these average results may be inaccurate given the fact that the field blank from day 2 was shown to be contaminated. The more reliable results from day 1 still proved to be exceeding regulatory limits, with total coliform concentrations of 3,130 mpn per 100 milliliters and E. coli concentrations of 173 mpn per 100 milliliters. The total coliform concentrations were shown to exceed the 2,000 mpn per 100 milliliters for Rec 2 use and the 200 mpn per 100 milliliter for Rec 1 use. E. coli concentrations were also shown to significantly exceed the limit of 126 mpn per 100 milliliters. Therefore, Lake Murray is confirmed to be under impaired status through these resulting concentrations and exceed regulatory maximum guidelines. However, it is also important to understand that this lab includes limited sampling due to time constraints. It is recommended that more sampling and testing occurs to accurately reflect the water quality at Lake Murray. As stated in the water quality objectives for indicator bacteria, Fecal coliform concentrations and total coliform concentrations listed in water quality objectives for indicator bacteria are based on many samples taken over a 30-day period, whereas this laboratory only analyzed a few samples over a week period due to time constraints. It is likely that data obtained from this laboratory may not be an accurate depiction of the water quality of Lake Murray, and therefore, it is suggested that future analyses are conducted following procedures as specified by the water quality objectives for indicator bacteria.
In conclusion, a groundwater sample from Lake Murray was utilized to test this theory. Contaminants and bodies of water were only recognized if there was good evidence of violations of the respective water quality goals. Water bodies were listed according to their placement with hydraulic limitations. The overall coliform and E. coli concentrations of Lake Murray were determined to be beyond regulations in a degraded condition by the Quantitre 2000 assessments. San Diego's Water Board specifies that Rec 2 users be restricted to 2000 mpn over ml and Rec 1 users to 200 mpn over 100 ml of total coliform bacteria per 100 ml of water. As mentioned, the overall coliform and E. coli concentration was beyond, and also the average E. coli concentration of 918.5 mpn over 100 ml was also shown to significantly exceed the limit of 126 mpn per 100 ml. Due to the fact that the field blank was found to be contaminated, day 2 results may be doubtful. The far more reliable is the day 1 results of 173 mpn E. coli over 100 ml and 3130 mpn total coliform over 100 ml. However, still exceeds the limits. As a result of these findings, Lake Murray has been confirmed to be impaired.
Hi, as for the recommendations, our group has a few advices that we'd like to share in regard of sample collecting. First, in the field, sample collectors must be wearing covering clothes, medical gloves, and closed-toe shoes for safety, because the water might be contaminated. In addition, collecting more samples than needed might be helpful for analysis. After collecting the water samples, the samples need to be carried to the lab in a clean water kit that is closed tightly. In the lab, samples have to be tested for contaminations. Furthermore, samples have to be restricted from contaminations to avoid testing errors. After testing the samples, washing hands thoroughly with soap after touching the water sample is crucial. In fact, the water samples need to be treated as hazardous waste in order to avoid risk to the human health. These are the recommendations that our group has performed during our experiment, and this is a picture that is used as a proof that the guidelines discussed in this section was practiced by our team members. Thank you.